thing واعمل ريست لحالك بحيث انه تاخذ معلومه وفكر بايجابيه ببين حل اخر شيء، لانه اعتقد الفاينل كان فيه علامات كويسه بالنسبه للسي في اس. هلا ما سنتكلم عنه للامانه هذا الشابتر يعطى للبوست جراد ستودنتس، ولكن انا اذا بتلاحظوا بالسلايدات اللي موجوده اصلا والسلايدات اللي عدلتها تم اختصار حوالي 50% من الموضوع والتركيز على اشياء مهمه جدا، لانه somehow it's advanced one بس لانه انتم medical students يعني advanced لطلاب البيولوجي بس مش لطلاب الميديسن. فهذا الشابتر بيحكي عن الهيموغلوبين and the inherited disorders of globin synthesis واشياء ذكرناها سابقا اللي راح نتكلم فيه عن الستركتشر genetic control synthesis of hemoglobin, classification of the disorders of hemoglobins and some examples thalassemias and these things. طيب هذا كله حكينا عنه. ايش الجديد؟ الجديد اللي هو الdiagnosis, how we do diagnosis, how we read the results of the hemoglobinopathies. Again, if you open a reference like this one راح تلاحظ انه الستركتشر اوف ذا جين از ديفرنت وراسم لك هون كروموسوم 16 كروموسوم 11 ف sometimes it's confusion للقارئ، ولكن حبيت اعرض هاي الاشياء بطريقه مختلفه لانه الريفرنسز او السكولز تطلع عليهم لما بتقرا كتاب بيعرض لك اياها بطريقه اللي هو بفهمها بالنسبه للباك جراوند اللي معطيها لطلابه. هلا لازم نتذكر دائما انه عندي على الكروموسوم 16 هنا مجموعه من الجينز اسمهم alpha gene family, and chromosome 11 we have beta gene family, and the combination between these subunits will give you these different hemoglobins. احنا يمكن حكينا عن جوور 1، هلا في عندي شيء اسمه بورتلاند hemoglobin, جوور 2 hemoglobin. هذا الكومبينيشن تبعهم somehow different from the previous developmental hemoglobins، وهذول كلهم بمرحله الامبريو اللي هو 3 months of pregnancy, and later we have shifting in the synthesis of hemoglobins. We have kind of regulation that rely on LCR sequences or enhancer sequences and silencers, how they control the gene expression to produce what's called fetus hemoglobin, and it will be shifted to the adult one besides the F1. Okay, this is just another look to the gene, and these are the major genes that we have in the adult period from the point of view of the size. إذا بتشوف هاي الكودز اللي هي أشكالها مستطيلات والوان والى اخره، اذا بتفتح الويب سايت شوي احيانا يعني بعض الناس تعقد منها لانه ايش الستركتشر هذا. It represents here the exons sites for example, and the beginning and the end of the gene, and they are color coded. It's just kind of اذا بدك شو بسموها بال graphic design, how they design the genes, how they draw the genes. بتروح على reference ثاني برسمها بطريقة مختلفة. But the same basic thing. Every gene consists of exons, introns, end and beginning. ما قدرش يتحمل محمد. فعشان هيك أنا فصلتهم إلى الألفا جين beta gene family and alpha gene family and the most important genes on the chromosome. ليش هذا مهم؟ لأنه أنا إذا بكرة بدي أعمل diagnosis, molecular diagnosis, I have to know the DNA sequence at that site مشان اعمل سيرش معين او ريسيرش معين اذا بدك على ميوتيشن معين على تشينج معين بهذا الجين. اجين زي ما حكينا قبل شوي انه في عندنا flanking regions اللي هم 5 prime region 3 prime region بسموهم على مستوى الار ان اي اذا صار له اكسبريشن على مستوى ار ان اي، and here we have the internic regions that have to be spliced in order to have the complete or mature messenger RNA and be transported outside in order to synthesize the globin as any other protein. So just remember that we have these sequences as extra sequences وهذول لهم function معين في regulation. بتلاحظوا هنا في عندنا stars موجودين اللي هي بتأشر لشيء اسمه البوليمورفيزمز، يعني في شخص عنده في هذا المكان A، في شخص ثاني عنده T, G. These are the sites of polymorphic nucleotides or sometimes mutations at these sites. فعشان هيك بحطوا عليهم الستارز هذول indication of the hotspot sites of mutations or polymorphisms. هلا هذول الميوتيشنز بتقل نسبهم لانه احنا بصير في عندنا splicing، يعني كل ما كان الميوتيشن موزع على الجين كامل after the transcription process you will have small portion of mutations. عشان هيك كل شوي يعني the percentage of the gene mutation will be reduced while we have the messenger RNA. However, sometimes you could inherit certain mutations and the type of the mutation depends on the size sometimes or the location of that mutation. هلا شيء already اندرس بالجينيتكس انه we have regulation process of the gene expression and we have what's called regulatory elements اللي هم sequences. كلمة elements هون تعني sequences of DNA موجودين على السلسلة تبعون الجينز او ما قبل او بعد الجينز. جزء منهم اللي هو البروموترز فور اكزامبل. البروموتر اللي هو المكان اللي ببلش منه التعرف على الجين وبعدين نعمل عملية transcription للRNA. We have enhancers. They are sequences could be far away from the gene but they play a role in the regulation of the gene expression. That's why they are called enhancers. العكس لهم بسموهم silencers إنه بيعملوا down regulation of the gene. And we have what's called locus control regions and they are very famous in the hemoglobin expression or hemoglobin synthesis. تمام اللي هو الألفا البيتا جينز هذول اللي لهم دور في إنه ليش أنا في عندي Goer hemoglobin, for example, and hemoglobin F, and so on. And we have also another name which is called locus activating regions because they activate the gene expression specifically in the beta globin family. مش بس البيتا gene itself. Alpha gene cluster is another kind of regulatory sequence, has homologous upstream sequences designated as HS40 اللي هو بسموهم homologous sequences. And they are DNA's hypersensitive sites. What does that mean? في الجين كسيكونس اوف دي ان اي في اماكن بيرتبط فيها بروتينز معينه لهم دور في ريجوليشن زي البروموتر على سبيل المثال. البروموتر بيرتبط فيه ترانسكريبشن فاكتورز بعدين بيرتبط الار ان اي بوليمريز اند سو اون. هلا بعض المناطق بتضل فري هاي ما بيرتبط فيها شيء. تمام. هلا عشان نعرف هم كيف اكتشفوا انه البروموتر هذا موجود هون وهذا البروتين بيرتبط في هذا المكان. It's just simple approach of mixing of the protein and the DNA sequence and then treating the mixture with DNAs. وبالتالي إذا كان في عندي سلسلة من الدي ان اي دبل ستراندد والبروتين ماسك هون والمناطق الأخرى فاضية الدي ان اي شو بيعمل؟ degrading. فبصير degradation لكل شيء ما عدا الدي ان اي اللي بخبى جوات البروتين اللي ماسك فيه. And after that we add deactivator عشان اعمل inhibition للDNAs. And then separation of the protein from the DNA. كيف بعمله؟ protease. بحطم البروتين بضل عندي fragments of DNA. I analyze هذول الفراجمنتس تبعون الDNA. This is يعني experimental approach how we think simple ideas could produce يعني excellent approach. فالDNAs hypersensitive sites معناها هذول الأماكن لا يرتبط فيها بروتينات معينة. They play a role in the regulation because they are protein unbinding sequences or elements. And when the regulation takes place, most transcriptional levels with post-transcriptional modification. What does that mean? Most of the time, the regulation is at the level of the transcription. And we have some regulation post-transcriptional or post-translational. هلا in fact بالنسبة للglobin story it starts when we have the hetero-euchromatin structure. كده بتتذكروا هذا الشيء. الكروموسوم عبارة عن DNA molecule مش الإكس. تمام؟ هاي شغلة مهمة. الكروموسوم عبارة عن DNA molecule طويل حسب نوع الكروموسوم مخلوط معه بروتينز اللي هو هيستون نون هيستون بروتينز وبيعملوا الستركشر اللي احنا بنشوفه. تمام؟ فالكروموسوم فعليا زي الليزر هذا الكليكر. تمام؟ هلا لما بصير في ريبليكيشن you will see the X shape بالنسبة للكروموسوم. 99% of the lifetime of the chromosome will be not like this. لا هيك ولا على شكل X. عبارة عن يعني سبونجي اور وول فورم اللي هو الشكل الصوفي للكروموسوم انه ماخذ تيريتوري احنا بنسميها بالخليه وحيز مكان في النيوكليوس ومفروض فيها هناك. هلا هاي هذا الحيز اللي هو كروماتين اريا بسموها عشان هيك احنا في عندنا مصطلح كروماتين. كروماتين بوصف الكروموسوم سواء كان بهذا الشكل او مفروض وفيه مناطق هيتيروكروماتين كير كوندنسد ومناطق يوكروماتين شوي مفروده they are active. الشاهد في الموضوع انه at that level we have regulation انه DNA compact as heterochromatin ولا شوي لوزي euchromatin. فكلمة at what level or when the regulation takes place starting from the DNA structure or the chromosomal structure until the end اللي هو post translational process. ولكن معظم الريجوليشن بصير على مستوى transcription and we have also post-translational modification. ايش post-translational؟ بعد ما صنع البروتين I could do certain modifications. Yeah. as a regulation process. هلا المنطقة اللي حكينا عنها اللي هو nuclease or DNA's hypersensitive sites are found at several locations in the beta globins, which means that they have a lot of LCR sequences and they are localized upstream of the gene. And I think this is mentioned in the regulation of our eukaryotic gene regulation in the genetics course. This key regulatory region established a transcriptionally active domain spanning the entire beta globin gene cluster. What does that mean? إذا بنرجع على الجين structure هون البيتا gene family, okay? على upstream site you will see LCR sequence which controls the expression of all of these genes. وبالتالي هاي ال LCR is responsible for the regulation all of these sequences. بس هذا ما بكفي. It needs another thing to regulate. And we have also several enhancer sequences have been identified in this cluster. So the regulatory sequence of the LCRs, they need also what's called enhancer sequences. And later they discovered that these regulatory genes bind number of erythropoietic or erythroid specific transcription factors, JATA1, NFE2 for example, or many other transcriptional factors. So the regulation requires the presence of LCR enhancer sequences and as well as transcriptional factor in order to regulate the gene expression. Thalassemia is an old story as a disease and it was described in 1925 as a first recognition as a disease from the point of view of the medical terminology, thalassemia, ولكن هي old disease for that. And they noticed that it's found in infants and they have anemic with splenomegaly phenotype. And after that, there was pathological description of the thalassemia itself, and it's known very well in the Mediterranean area. And the Greek name of that disease the name of the disease came from the Greek word وبعرفش ايش هاي باللاتيني معناها ولكن in English it means that the sea اللي هو الثلى وسيميا معناها انه something or something related to the blood itself. هلا الفريكونسي العالي reflects heterozygote protection against malaria in certain areas. It means that انه due to the invasion of malaria there is an increase in the frequency or incidence or prevalence of the thalassemia and even the other hemoglobinopathies in the Mediterranean area. And here we can see the distribution of the hemoglobinopathies in the old world, starting from the hemoglobin S, for example, hemoglobin C, and the other subtypes of hemoglobins and the beta thalassemia. We can notice that the area that is dominated by malaria is also dominated or has high frequency of the hemoglobinopathies as a kind of, it's not protective measure as much as exclusion of that population from being infected by malaria. So it's, again, it's not protective. It's just kind of, if you would, natural selection of those individuals. Thalassemia, as we mentioned before, has classified actually to alpha and beta, and we could have other types depending on the type of the mutation. The most common types اللي هم الالفا and البيتا. هلا البيتا more common because we have only 2 alleles for that one. The alpha because we have 4 alleles, it means that it's less dominant than the beta one. In another word, beta is more إذا بدك lethal أو more frequent than the alpha one. According to that, we have either alpha note, means that you have absence of that gene. Alpha plus, it means that there is expression of that one. This sign means there is a deletion. And the T means that there is no deletion at that area. There is a mutation. And there is a difference between deletion and the mutation in the gene itself. So we have different gene codes of the thalassemia genotypes, if you would, and phenotypes. For example, we have delta and beta thalassemia. It means that we have absence of the delta and beta subunits, delta 1, gamma 1, and so we have other subtypes or minor types of thalassemias that are not clinically significant in the population, but you could find some individuals with these kinds of thalassemia. The most important thing, it's Mendelian recessive inherited disease. It means that in order to have the disease, you have to have both alleles defected in the individual. ما بدي أقضي وقت كثير على beta thalassemia. It's very well known why we have beta thalassemia. But from the point of view of the mutation, مش بالضرورة يكون deletion في الجين itself. It could be the transcription itself. There is defection or defect in the transcription. Maybe the promoter area is defected. Maybe the gene or one of the exons in the gene is mutated or deleted. During the processing of the messenger RNA, again, going back to the gene expression. So every time we have a chance to get a new mutation, could be during transcription, could be during the processing of messenger RNA, in order to have defected proteins. So in the whole pathway of the transcription or let me say expression of the gene, we could have source of mutation that can lead to the disease itself, مش بس thalassemia, any other genetic disease. At the translational level also we have causes that will be related to the thalassemia, or maybe post-translational instability of the protein, which means that if you synthesize the protein, sometimes it could be unstable and it will be degraded by other proteins. But most of the time, the thalassemia is associated with the gene itself, if it's mutated or not mutated, besides the other types of the mutations. عشان هيك احنا بنركز دائما على الجينز themselves. بالالفا ثلاسيميا او بيتا ثلاسيميا we are talking about the alleles and the mutations in these alleles, and this is for example a description of what's called thalassemia trait. Alpha no thalassemia trait means that we have absence of one of the or both alleles on the same chromosome, and this is also similar one, and this is the meaning of having recessive disease or Mendelian inheritance disease. And here we will develop اللي هو البارت hemoglobin and hydrops, for example, and it will be lethal one, stillbirth individuals. This is how it's gone with the other individuals if they have, for example, alpha thalassemia trait, and here we have alpha thalassemia, but one of the alleles is affected, not both. Okay, it means that we have expression of one of the alleles in one of the chromosomes, and these are the possibilities of the individuals that will be, or offsprings that will be born in the future. And of course you will have hemoglobin H in this case because there is low percentage of hemoglobin alpha, or sorry, globin alpha compared with the beta that should be available for the hemoglobin synthesis, and that's why we have hemoglobin H as precipitate. And as a pathological diagnosis, you will see Heinz bodies as well as hemoglobin BART in these individuals. Because it relies on the percentage or percentile of the expression of these genes. Again, something repeated, okay? Sometimes they describe thalassemia as beta thalassemia intermedia, for example, which means that you have mild condition, or that is associated with homozygosity of the mild beta thalassemia. It means that you have one of the alleles is affected. That's why it's called mild one or trait, if you would. And the major one means that you have both alleles are defected. هلا موضوع الثلاسيميا is long story، وانا شلت سلايدات كثير بالمناسبه خاصه بالثلاسيميا، لانه if you dig down in the molecular biology of the thalassemia, sometimes you have to go back to the basic molecular biology to understand how the genes look like and how they are expressed. Okay, so that's the problem when we talk about thalassemia. That's why we have to take just superficial look to the mutations. اهلا دكتوره، كيف حالك؟ هلا the other thing is the other diseases. متى قديش ساعة بالله؟ بعد 10 دقائق يعني؟ من الآن؟ طيب خليني أشوف بس. اوكي فاين. اوكي يبدو انه في اجتماع الان لكم مع العميده.